So hello, everyone. Welcome from Lucca, or more precisely, Jocko, in Italy. I'm here at the Gordon's conference for the biology of aging. And today I'm presenting Omniscope's Immunage algorithm, which is part of our osLifetime application. Just a quick introduction. My name is Holger Heyn. I'm Chief Scientific Officer and co-founder of Omniscope. And at Omniscope, we see what your immune system is seeing, and I will tell you more in the next few minutes what I mean by that. So at Omniscope, we've developed technology that is deeply reading into the immune system, which, as you all know, has been perfected over the last 500 million years to detect and fight diseases. And what you see actually on the screen is a T cell detecting a diseased cell, in this case, a cancer cell, and killing it. And this is happening every day, keeping us healthy. And at Omniscope, we developed technology to read this one cell at a time. In more detail, we have developed technology that reads deeply into the immune system, specifically the T cell and their T cell receptors. So we sequence millions of T cell receptors per patient at time points, which really allows us to dynamically tracking the activity of the immune system when, for example, a patient is responding to immunotherapy or to a cancer vaccine, but also obviously we can quantify the process of T cell aging. This is a topic of the presentation I will be giving today. At a company, though, we have gone already a step further, looking at the biobanking of healthy immune cells. And we have engineered cells already, so T cells and the T cell receptor to detect and fight tumor cells. And this is something we have recently published in the Annals of Oncology in the pediatric immuno-oncology setting. But today I will very much focusing on our model of Immunage. So to model Immunage, we're looking at the T cell receptor sequence. So this is that specific part of the T cell receptor recognizing these diseased cells. Those T cells will get activated, they start dividing, and this is a natural amplifying signal we're picking up. At Omniscope, we have developed technology called OSTCR that reads millions of these T cells, and that really allows us to train a model of age using chronological age as an outcome. And with this, we can now use Immunage or immunaging to look at correlates, for example, T cell phenotype activity, activity states, or we can look at correlation to external metrics such as exercise. This will be the focus of the talk today. But we can also look at different interventions in the space of longevity, for example, nutritional changes or supplements and therapeutic interventions. But Immunage is only kind of one feature that we use to generate a more holistic model of age. So we use our proprietary sequencing methods to look at Immunage, protection score, inflammation, and immune tracking. And these features are integrated with more classical assays such as senescence, telomerase, or methylation clock assays. And together, we build a model of age, which we visualize through a web application, which initially starts with a similar avatar, together summarizing those different aging-related features. But then we can kind of scroll through the different features separately: the immune age, protection score, inflammation, senescence, telomeres, methylation age, and classical blood test markers. We need to look at our own profile and the evolution of that profile over time. As mentioned today, I will mainly focus on Immunage, and basically what you see on the screen is my own Immunage tracked over the last 2 years. And you see it's quite consistent, but there have been episodes of acceleration, of deceleration of Immunage throughout the last year, and I will tell you more what happened during those days. At very high level, we can integrate a digital biomarker, for example, here, Apple Watch data calculating the VO2 max. And we see already that a decrease in VO2 max is here related with an increase in biological or immune age. And I will talk more about this in the next slide. But I would like to kick it off talking about reproducibility. So we, besides performing replicates, we have been looking at repeated time points with very short distance of sampling. So here, a couple of samples were profiled with a week of sampling distance, and this really allowed us to look at the tendency of these replicates over time. And you see in the surface areas. Those are very much following the evolution of the immune age during those weeks. We're also looking at the classical replicate experiment where a single sample has been measured 3 times in this case. The immune age predictions are quite consistent with a very low standard deviation across the 3 replicates. And let's now zoom into this more specific period of variable immune age. This was a time where I've been suffering from Lyme's disease, and you see that the actual disease has been related with a deceleration over the lower immune age initially. And then during therapy, which was high-dose cyclodoxin treatment, we've seen an increase of the immune age going back to baseline. And immune age is one feature we're measuring, but we can then look at correlates, for example, immune phenotypes. So here we've seen a strong correlation, for example, with CD4 T cell activation. So within decreased immune age, we also see a decrease in CD4 T cell activity. And this gives us the first hint what immune age is summarizing from the cellular biology slide. But obviously, away from the single individual reviews and the personalized view of tracking immune age over time, we're now looking at larger cohorts tracked over time and start more systematically looking at correlatives, not only the cellular phenotype space, but also looking at external correlates. And this is one application I would like to highlight today. We look at correlation with mobility and exercise metrics through a collaboration with the Football Club Barcelona. And today I will specifically focusing on the first female team. So this is data collected over the last season where we've been sequencing blood from the first female team at different time points. And you see that in general, the immune age prediction is more or less correlating with the player's age, but then indicated by the errors through repeated sampling, we see quite variability happening over the course of the season. At very high level, this variability correlated with the number of games played. So players that played more matches, more games, showed an increased immune age over the course of the season. But then we looked much deeper and much more into detail where those players are positioned on the pitch. So what is the role of those players, being a defender, forward, goalkeeper, or midfielder? And this is summarized on the left, but on the right, we've broken that down in players that decelerate or accelerate immune age over the course of the season. And you see clearly that on the very right side, the players in green, so the forward players, all showed acceleration of immune age over the season, where most of the rest of the team showed a deceleration. And that really gave us a first hint that there's a difference in mobility that is related to accelerated or decelerated immune age. So we looked closer and we looked at GPS data. So GPS data is tracking the players' mobility or movement patterns across the season and matches and training. And probably those players are the most monitored people, or among the most monitored people on the planet. So we had a rich resource of GPS data, and we started correlating the decrease or increase of immune age with GPS metrics. And what we found is there are specific metrics, for example, high-speed running, measures or high metabolic load distance that clearly correlated with an increase of immune age over the season. So with this, we now start disentangling what is happening on the outside world, so external load of the player measured by GPS data, and how much that is impacting the internal load. And with this, we are calculating a total load of the player, but also across the entire team. Here shown for the women's first team. And for a medical staff or a coach, this is the first kind of warning signal that the immune profile of the team is going into a different direction, potentially associated with injury risk, but we can also zoom into specific players' profile and have a more personalized view, again, overlaying the inner load features such as immune age together with GPS matrix, as shown here for immune age and inflammation. And with this, I would like to wrap up. I hope I could have convinced you that T cells are a natural sensor. We can use this sensor to track immune aging over years, but also in a shorter time frame, for example, looking at interventions such as exercise, nutritional patterns, or even disease. Thank you very much for your attention.